Hi there. In this video, I'll be talking about eukaryotic genomes. And from the last videos, you'll understand that eukaryotes really are a blended genome. So there's a nuclear genome, there's a mitochondrial genome that originated from alpha proteobacteria, and then in the plants, there's a mixture, again, of a genome from cyanobacteria, and with those two, there are horizontal gene transfers taking place. And then in protists, there are often, a red algae, a green alga that have become an endosymbiont and an organelle. So all those genomes, the nuclear, plastid, and mitochondrial genome from the red and green algae have joined in there. So eukaryotes are really a mixture of different types of essentially prokaryotic organisms, bacterial and archaeal, that have blended together into larger and larger genomes. They've also increased their number of things like transposons, introns, and repetitive elements, and have a lower percentage of mRNA encoding genes in general than do bacteria and archaea. So on this genome size graph again, you can see we've already been through the viruses, organelles, and bacteria and archaea, and then we come up here to eukaryotes, which essentially are genes that originated back here and have been blended together and elaborated upon. But also up in the eukaryotes, there's been an increase in intergenic sequences, so that, by the time you get to mammals and some of these organisms, much less than 10%, sometimes less than 1% of the genome actually consists of mRNA genes that encode proteins. So, as I said, they're blended genomes. So you have this group here, the eukaryotes, which came off of a branch of the archaea. And so they have an archaeal genome, and then all of these little arrows coming over here of the endosymbionts that moved over to become different organelles and contributed to the total genome of the eukaryotes. And it's even more complex than that, as I indicated in the previous video. The nuclei come in here, probably from planktomycetes, joining with an archaeal cell, and then you get the mitochondria added, the plastids added, but then there's all of these endosymbiotic events that, again, blend even more genomes with other genomes. So it's a eukaryote inside of a eukaryote, and eventually a eukaryote inside of a eukaryote inside of a eukaryote, and inside of each one of those eukaryote cells, you have at least a nucleus and a mitochondrial genome, and some of them, of course, had a plastid genome as well. So again, these are blended genomes in eukaryotes. So I will start first by talking about Arabidopsis thaliana, which of course is a plant, a higher plant. So it has a nuclear genome, a mitochondrial genome, and a plastid genome. And it was first used as a genetic model for plants starting in the 50s and 60s, and it's moved forward since then. So we know a lot about the genetics and all of the mutations in the Arabidopsis thaliana plant. Also, it has a very small genome, so that made sequencing a little bit easier. It's a much smaller genome than the human genome, so this was not a bad place to start for studying plants. It's a four-parted flower. Here's how development occurs. So it's got all the complexities of an angiosperm, a higher plant, but a fairly small genome. So it can be sort of genetically and molecularly dissected to study different functions of the genes and their gene products. So here's a bit of a breakdown of the genome of Arabidopsis thaliana. It has 157 million base pairs in its genome, which, compared to the bacteria I was talking about, is from probably 10 times to over 50 times the size of a bacterial genome, so quite a bit larger. 27 to 31,000 genes that encode 35,000 proteins. And so you can see already from this that there are more proteins than protein coding genes. And that's because there's alternative splicing. So a single RNA molecule, mRNA molecule, can be spliced in different ways, 2 or 3 or 5 different ways to produce different proteins. And so that's the first time you start seeing this to a large extent in multicellular organisms. So a lot of metabolic processes here, about a quarter to more than a quarter, are in basic metabolic functions. And then transcription, DNA replication, and translation take up smaller proportions of the mRNA genes. So now you're getting more specialized functions than what were in bacteria. A lot of specialized functions. And the basic metabolic functions, although there are more genes, they take up less of a percent of the genome. But you can also see a lot more genes for developmental processes because it's a multicellular organism, and responses to stress and external stimuli. So plants sit out there in the wind, in the sun, the winter, the summer, and they have to be able to change their response to those stimuli and conditions by shifting gene expression to things that will help them withstand strong winds, withstand cold temperatures, hot temperatures, too much sun, too little sun, etc. So this presents a partial analysis of the genome of Arabidopsis. You see that the total here isn't quite 150 million base pairs. It's only 102. So people are still going through the genome of the sequences of this genome. But it has 5 chromosomes, and here's the approximate length of each chromosome and the number of genes spread out among those chromosomes, and then the number of proteins produced by those genes. And you can see in all cases, more proteins are formed than genes, again, indicating alternative splicing. And then there's the mitochondrial genome and the chloroplast genome. And next up here you see that there are a lot of ribosomal RNA genes. And this is true of most eukaryotes. They carry a large number of ribosomal RNA genes, usually in excess of what they need. So in general, they probably need anywhere between 100 and 300 ribosomal copies, but they have excess of that in this case. 689 tRNA genes. Again, they will have different versions of the tRNAs for each of the amino acids, and some codons are used more often than others, so they're needed in different cells in different proportions. And then there are 71 snRNAs. These help to process the ribosomal RNA, but also other RNAs in the organism, in the nucleus. So now we've got more RNA processing going on, and then there are microRNAs that control the expression of the RNAs already formed, and so there's more control also here. Part of those are response to external stimuli also, so those are some of the things that they're causing. So a lot more small RNAs and a lot more alternative splicing of the RNAs. So more variability beyond just the genome. Also in eukaryotes, including Arabidopsis, there are many cell compartments. So this pie chart is showing where those proteins that are encoded in the nucleus primarily, where are they going? Which compartment are they going to? So About 8.8% of them are headed towards the chloroplasts, and another 3.5% are headed towards plastids in general. That is, they could be amyloplasts or elioplasts, etc. Some are winding up in the cytoplasm. Some are winding up on membranes. Some are going directly to the plasma membrane, 5.8% to the nucleus, 2.7% to the mitochondria, and then some to other intracellular components, mainly membrane components, and then other places as well that are unknown. So 31% we don't really know, or at least from this analysis, it's not known where those were ending up in the cell. So arriving at those destinations means that many of the proteins have a signal sequence or a transit peptide on their amino ends and a few on their carboxyl ends to wind up in those different compartments or attached to ribosomes or wherever they're headed. So this requires another level of complexity that isn't found in bacteria. So bacteria have essentially two places that those proteins can end up, either in the cytosol or in the membrane. And if they have two membranes, then there are a couple of other places they can wind up: in the periplasmic space, in the outer membrane, the inner membrane, or in the case of planktomycetes, there may be several other membranes where they might end up. So they would have to have a transit sequence on those as well. But for eukaryotes, there are many, many more membranes and locations and molecules or macromolecules where those proteins have to end up. The second plant I want to talk to is rice, Oryza sativa. It's also got a small genome, but it's economically much more important than Arabidopsis thaliana. Rice is commonly eaten by more than half of the human population, and about 20% of the total calories eaten by people in the world comes from rice or rice products. So it's quite important to the human population. And there are many, many varieties of rice. Long grain, short grain, there's darker grains, lighter grains, and so many, many different cultivated varieties of rice. And as you can see here from the plant, it looks quite a bit like any grass, and so it is a grass. It looks sort of like wheat, and it's related to wheat, but not real closely related to wheat. But you can see clearly that it is a grass within the family Poaceae, which once was called Gramineae, but it's changed. It has 12 chromosomes and is about double the size of the genome of Arabidopsis thaliana, with a large number of genes also, 61,600, encoding more proteins. So there's also alternative splicing going on here. You can see it's only partially been sequenced. This last chromosome 12 is not complete yet. And there's the mitochondrial genome and the chloroplast genome. It consists, as I said, of 61,600 genes, including hundreds to thousands of ribosomal RNA genes and 738 tRNA genes. The reason I say several hundred to thousands of ribosomal RNA genes is because that's variable, even within a cell or a plant. It's quite variable. The species within the grass family are quite variable too, so they feed a lot of the human population. Rice has the smallest genome, and as you move up here's Zea maize, Triticum aestivum, which is wheat, and the largest genome here is Avena sativa, which is oats. So quite a variation in genome size from down here around 300 million to more than 10 billion nucleotides or base pairs. And humans have about a 3 billion base pair genome, so this is even larger than the human genome. And here are those grasses, starting with rice. This one has a 483 million base pair genome, although some rice is down in the 300 million range. Millet at 490 million base pairs. Sugarcane at 1 billion base pairs, 1.6 billion for sorghum. Maize is at 2.5. Wheat is at about 6 million base pairs, about twice the size of a human genome. And then oats have a genome size of around 11 billion base pairs. Now, although they have large differences in their genome sizes, their coding sequences, that is the mRNA genes, they have about the same content of those. So all of the extra are other regions, intergenic, highly repetitive sequences, including transposons. When the chromosomes are aligned, many of the regions are syntonous, that is, many parts of the chromosomes are syntonous, even though they have differences in chromosome number. So starting with the smallest genome in the center, which is rice here, then you can see the chromosomes there, 1, 2, 3, 4, etc. And they're lined up with the chromosomes of millet, which is the next one out. And you can see for the most part, they're syntonous, although the chromosomes in some cases have joined together with other chromosomes, so the chromosome number is somewhat different. Also, some regions have moved around on the chromosome, so you get these movements of large sections of chromosomes and sometimes inversions also where they switch directions. As you move further out on these rings, the next one out is sugarcane, and you can see here a large deletion of a large section of one of the chromosomes. That turns out not to delete too many genes because, as I said, most of the chromosomes are made of what are called intergenic regions that don't encode any mRNAs and proteins. So going further out, you can see more and more changes, parts of chromosomes fusing, parts of chromosomes missing, parts of chromosomes moving from one place to another, chromosomes fusing, chromosomes splitting. But overall, the gene content is about the same. The mRNA gene content is around the same for all of these. And remember from a lecture before, the maize genome itself, 84% of that genome is made of transposons, and most of the grasses have a similar content of a very high number of transposons, a smaller proportion of mRNA genes, although they generally have more mRNA genes than, say, mammals, including humans. So we have approximately, 23 to 26,000 genes. And these grass genomes have around 50 to 60,000 genes. But then there's not as much alternative splicing as there occurs in animals. So there's been a sort of a difference in the way that they go about encoding and making mRNAs. Then about 1% is made of microRNAs, and then another 10% made of other things, including long introns. And studies have shown that if you look at the chromosomes in maize, and this is just one example of one chromosome, that the center portion of each chromosome is made up of, in this case, LTR retrotransposons, which are the ones that are in the highest number. And the genes, the mRNA genes, are out towards the ends of the chromosomes. And for maize, this is true of all of its chromosomes. It's probably true of the other grasses. It's just that I haven't seen the data on that. This is a comparison of the gene families, or essentially genes, that are in common or unique to each of these species. So rice, maize, and sorghum are the three the three grasses, and it doesn't include all of the genes. So there's about 50,000 to 60,000 genes total in each one of these. So it's just the genes that were compared in this study. And then Arabidopsis is not a grass, it's a eudicot. So in general, though, between all four of these, a large proportion, 65 to 83% of the genes are in common about 65 in common to all, and 83% are in common to all the grasses. So that, as I said in that previous one, if you look at the synteny of all of those genes, you can find comparisons of large tracts of each one of the chromosomes that are similar in each one of the grasses. And by the time we get back to Arabidopsis, some of that has broken down, but you can find many of the same genes in Arabidopsis or any plant as well. So the way that you can look at this is that about 1,000 genes are unique here, and in each one of these 1,400 and 200. So for these two, fewer are unique, and there's more in common, many more genes in common to both. And then by the time you get down here, only 81 genes are in common with rice and Arabidopsis together, so a small number. And this is the one in common with, or that are in common only with maize and Arabidopsis. And this number is in common only between sorghum and Arabidopsis. So you can see there are many, many more common genes among all of those than there are unique genes or genes that are common to just pairs of those organisms, especially when you compare the grasses with Arabidopsis, which is the eudicot. The next species I want to talk about is a fungus. It's Saccharomyces cerevisiae. The reason I picked this one next is it's closest to animals. So it's part of this group called Opisthokonta, which includes fungi and animals together. Saccharomyces can do a lot of different things. Here it is. It's a budding yeast, so it buds out of one end and forms a new cell, which is a little smaller than the mother cell. Here it is growing on a plate. You can use it to ferment sugars to make beer, wine, and bread, mainly to produce CO2 here for the bread for leavening. But it can go through a diploid stage where it divides off diploid cells every time, or it can go into a haploid stage where it divides off cells that are all haploids. And to go from a diploid stage into a haploid stage, it undergoes meiosis, producing haploid spores, and then going through the haploid division stage. Or it can join two cells together and form a diploid cell and then go through diploid mitosis. It can also undergo apoptosis, which is important to study that in terms of apoptosis in human cells. And it can form multicellular pseudohyphae as well. So many different things, many different life processes it can go through. Its genome is about 3 times as large, 2.5 times as large as an E. coli genome. And about the size, a little larger than a planktomycete genome, but then about 10 times smaller than a plant genome, so that's the range. And it has about 4,862 protein coding genes. So it does have a portion of those assigned to energy metabolism, DNA metabolism, as well as replication, transcription, translation, a lot on RNA metabolism, amino acid synthesis, etc., and then response to stress and the environmental conditions. So mainly people use it under anaerobic conditions to go through fermentation to produce alcohol or CO2 or both. And it can also grow aerobically as well. There also is a portion of the genome assigned to organelle. organization to creating the cell envelope, which includes a chitin cell wall, and control of the cell cycle. And as you could see, it can undergo haploid mitosis, diploid mitosis, meiosis, and so control of those functions as well. So here is a karyotype, the chromosomes, from 1 to 16, the chromosome number, and then the mitochondrial genome here, a 12 megabase genome, and here are the sizes of each one of the chromosomes, ranging in size from about 230,000 to over 1 million base pairs. For the largest chromosome, it's actually about 1.5 million base pairs. So there are the genes, the proteins formed from those, and you can see the numbers almost exactly the same. So there's not a lot of alternative splicing indicated here, if at all. The tRNAs here are numerous, almost 300 of those, and spread out throughout the chromosomes, whereas the ribosomal genes, about 140 on average, are only on chromosome number 12. And these chromosomes can actually be separated by special techniques on agarose gels, so you can separate these. But chromosome number 12 here was always a bit fuzzy. That means that there were multiple versions of chromosome 12, and it turns out that 140 for the ribosomal copy number, ribosomal RNA gene copy number, is the average. But there are some versions that are larger than that, some versions that are smaller. So the chromosomes are sort of showing that, in that they're of variable length because of those variations in ribosomal RNA copy number. Also, when this genome was sequenced, it was difficult to get through that ribosomal region because it consists of repetitive elements. And anytime in sequencing you have repetitive elements, you're sequencing little sections and then trying to piece those together. But if you have multiple repetitive elements, it's hard to tell where the repeats begin and end because there are so many of them. And so most of the time in these sequencing projects, they get to a repetitive region, a region of highly repetitive sequences, and they sequence up to one end, and then they start sequencing from the other end and kind of give up on the middle portion. One of the next genomic projects that was completed was the sequencing of Cinorhabditis elegans, which is a nematode. And just like Arabidopsis and E. coli and Saccharomyces cerevisiae, it had been used for many decades for genetic studies, looking at genes and how development occurs and other capabilities of each one of those organisms, and sort of dissected them genetically before they looked at the genomic sequences many years later. So here are some individuals of Cinorhabditis elegans. It's about a millimeter in length. Actually, the male is a little shorter than the hermaphrodite. There are two sexes. One is a hermaphrodite that forms both eggs and sperm, and then fertilizes those internally and lays eggs that already contain the zygote, so they're already starting to develop. And then males that don't have any egg producing and egg laying behavior, but they form sperm. It's about 1,000 to 1 ratio between hermaphrodites and males, and so mating is very infrequent in this species. So that's one reason it was chosen too, because it was an easy organism to sort of dissect developmentally and behaviorally as well. So you can see down here some of the developmental stages. So you could actually follow the cells through the entire developmental process. And when this was done, then you could look at a cell fate map. So this is following each one of the cell divisions from the zygote on forward through the embryonic stage and then the post-embryonic stage. And in every case, you found that C. elegans adults had 959 somatic cells, including 302 neurons, and then eggs and sperm are extra because they're being produced all the time. So they knew that by following these cell divisions that this cell would always become that type of cell, and the cell down here would always become that type of cell, and so on. But they also noticed in some cell divisions, indicated by some of these arrows, that during cell division, one of the cells always died, and the other cell went on to become something else. And so they started looking at this as a normal part of development, and eventually it was called programmed cell death or apoptosis, so that these cells were programmed to die by a certain cell division pattern at a certain time, which then allowed the other cell to survive and become a specialized cell of one sort or another. We now know that this is pretty common all the way through animals and plants and fungi, and that there is something called programmed cell death or apoptosis that occurs at different times. Some are during development and others are later. For instance, there are sort of proteins that determine how much damage there is in a nucleus, how much DNA damage, and they'll signal p53, which is a protein that signals other parts of the cell, other proteins in the cell, to kill that cell, to undergo programmed cell death. So it's a valuable process for development and also in protecting the total organism, especially multicellular organisms, from cell damage of some cells. Here is one example of that. So here are some cell divisions that have been mapped out on this Caenorhabditis elegans and where they form. And you can see in some of these divisions, one of the cells marked by an X dies, and in other cases it survives and becomes part of the organism. So in essence, some of these cells here, you don't want that type of cell down at this end of the organism or that end. And so in those cell divisions, those cells die, they're killed. Another one here is a programmed cell death of certain neurons that have specific functions in the male versus the female, or the hermaphrodite. So in this case over here, it's a CEM, or a male-specific chemotaxis neuron. So the male needs to be able to sense where the hermaphrodite is for mating, whereas the hermaphrodite doesn't need to sense where the male is. She can produce her sperm and eggs, so she doesn't need to find the mate. It's the male that needs to find the mate. So in this one cell division, in the male, there's a cell division where both cells develop. One develops into the male-specific chemotaxis neuron. And in the hermaphrodite, that neuron isn't needed because she's not trying to find a mate. The male is doing that part. So during that cell division, that cell dies, and the male-specific chemotaxis neuron never develops. On the other hand, the hermaphrodite lays eggs, so the hermaphrodite needs to have innervate the muscles, which will then push the egg out, whereas the male doesn't need to push the eggs out. So that neuron innervating that particular muscle isn't necessary. That cell dies and it's never formed. In the hermaphrodite, that goes on to form the hermaphrodite-specific egg-laying neuron. And those are just two examples in this tiny little animal. There are many more examples in this animal as well, so that this isn't all that happens. And this happens to a larger degree in larger organisms with more complex developmental patterns as well. So here's a pie chart of Caenorhabditis elegans, a genome size of about 100 million base pairs, 20 to 25,000 genes, which is about what we have also, by the way. General functions, there's DNA metabolism, transcription, translation, and those things. But one of the large parts of the genome is in signal transduction, and that's taking a signal from the outside of the cell and then transferring that inside the cell and then signaling different proteins and different functions inside of the cell. So that's really become a much larger part of the genome in animals. Their cells have to respond to stimuli, to hormones, and other things, temperatures. And so that all has to be transmitted into the cell, and then the cell expression levels usually change dependent on those signals. So here are the chromosomes. There are 5 chromosomes plus the X chromosome. Females have 2 X chromosomes. Males are X0 as far as their amount of their sex chromosomes. About 100 million base pairs of DNA, 25,000 genes with 26,000 protein products. So there's quite a bit of alternative splicing. There are an average of 275 ribosomal RNA genes, but that's an average again. 590 tRNA genes, 120 microRNA genes, 70 spliceosomal, and 30 SNO RNA. So a lot more of these small RNAs that are controlling different functions and processing different RNAs within each cell. The next genome I want to talk about is that of Drosophila melanogaster. This was one of the first genetic models for animals that was used, and it's been studied since the early 20th century, so a long time. So studying chromosomes in Drosophila, genetic patterns, mutants, even cancer. So down here, some of these genes down here that control different functions are also known in human cancers, so you can use Drosophila as a model for that. It's also used as a model for development and developmental genes, including Hox genes, homeodomain genes. And it's been used to study chromosome structure, especially these polytene chromosomes, which are a product of replication of the chromosomes over and over and over again without separating the replicated chromosomes. So you can see they form these large bundles of chromosomes. And these homeotic genes over here control many different functions, but this is one called bithorax, where the flies have 2 sets of wings and thoraxes. And here's an additional one down here. This is called antennapedia, where instead of having antennae on the head location here, feet are formed there, and that's caused by a mutation in one of the Hox genes. And again, analogous mutations occur in humans and other mammals. So the study in fruit flies first was helpful for looking at those homeodomain and homeobox genes. So here are the polytene chromosomes, and then they have studied which ones are being turned on and turned off by looking at these puffs. And here are some eye mutants over here on the right. These ones on the upper right are in alligators and other organisms looking at development based on the Hox genes. And as I said, these types of genes were first discovered in Drosophila, but then subsequently discovered in vertebrates. Here is the life cycle of Drosophila. So here's the adult fly over here on the left. And then here's the egg cell from the female and the sperm from the male. Note that there already are some gradients of proteins and other components, RNAs and proteins, from one end of the oocyte to the other. So there's already a difference from one end of the egg to the other. And then you get the zygote. So now you have a diploid nucleus consisting of the male and female genes. And then there are 9 nuclear divisions without cell division. So by that time, there are over 500 nuclei. And then shortly thereafter, those nuclei then migrate to the periphery. Cell membranes form around those. So now you have sort of a hollow ball called the blastoderm. And then you start getting differential expression of genes from one end to the other, sort of defining the both of the ends and the middle in different ways, just molecularly. So the first developmental genes to turn on are the gap genes, and then the pair rule genes start setting up a segmentation pattern between one cell type and the other, and the next cell type and the other. And then there are segment polarity genes, which further bisect those segments. And then you start getting expression of these homeotic genes, which start turning on. The larvae start feeding in 3 different stages, and then it pupates at the end. And within the pupa, then, the organism is undergoing reorganization. So there are things called imaginal discs that are turning on each one of those sets of genes that will form different body parts in the adult organism. And in a way, it's feeding off of some of the cells in there to produce new cell types that will form the adult organism, including the head and the antennae and the legs and the wings, all of the different body parts, and putting them in the proper location on that organism. So here's the karyotype, or the chromosomes here, of Drosophila melanogaster. This is the chromosome 2 left arm, chromosome 2 right arm, 3 left, 3 right, and chromosome 4, and then the X, and then there's a small Y chromosome as well. So the total genome is about 133 million base pairs. There are each of the sizes of the chromosomes and the number of genes on each, plus the number of proteins that can be formed. So you can see there is some alternative splicing going on so that you have more proteins than you have genes, because some genes can form more than one mRNA, which encodes more than one different protein. 100 to 240 ribosomal RNA genes. Again, these are variable. They have actually made mutants down to 15 copies. And when you get down to 15 copies, development stops. The minimum to create a Drosophila, even though development is slow, is around 20 to 30 copies. And 100 is usually the minimum in a wild population. Then there are 297 tRNA genes. Again, they are expressed differentially in different types of tissues. And then greater than 15,000 non-coding RNAs. So now you start seeing in animals where these non-coding genes are becoming a very large proportion of the transcripts. And the number of mRNAs becomes smaller or equal to the number of non-coding RNA genes. So the study of Drosophila has helped in the study of other organisms as well. So this is just looking at when those genes turn on, or where they turn on in the at different stages of development. This is a normal Drosophila, and then this is a mutant. So you can see some of the things don't work quite as well. And this shows a sea urchin, nematode, fruit fly, that's Drosophila, zebrafish, frog, and mouse, and human would be similar to this, showing when some of those genes are turned on during development. And although the timing is different in each one of these, some of the same genes are turned on at the same time during a portion of development, even though in some of these you can see that you start getting more and more complex organisms formed in each case. So in Drosophila, there are several different types of genes and the proteins that are formed from those. So there are, in the egg, there are maternal products, mainly BCD, NOS, CAD, and HB. HB is hunchback, and BCD is bicoid. So these genes are differentially expressed from one end to the other, and the proteins that are translated are also differentially distributed in that egg. So there are already changes in the egg that set up the initial pattern of development. And BCD, CAD, and NOS affect one another. So NOS will inhibit BCD, BCD will inhibit CAD. But then when you get down to the gap genes, different genes turn on and make different proteins, but also the CAD, BCD, and NOS can affect those. So CAD will turn on the GT gene, BCD will turn on both of these, HB and GT, but NOS will inhibit HB, so it's at one end, HB is at the other end. This sets up sort of a head versus tail versus middle, then you get sort of a finer distinction between one end and the other. And remember, by this time, you have over 500 cells. And then the parallel genes turn on later, and they then set up this segmentation in the animal, and early and late, more and more segmentation. Then segment polarity sets up poles, polarity of each one of the segments, the head end versus the tail end. And then finally you get all of these leading into this down here, which are the homeotic genes that then turn on during pupation. And then that will put the head where it has to be, put the eyes where they have to be, put the antenna where it has to be, and the legs where they have to be, etc. Not only the external organs, but everything interior, the muscles, the nerves, the stomach, etc. And although I haven't shown it, what these Hox genes are doing is they are turning on large sets of other genes so that they don't form the leg, they turn on the genes. So they're transcription factors that turn on the genes that form the legs, to form the muscles and the nerves and things like that. And they don't form the wings or the thorax, but they turn the genes on, a large set of genes, to build a thorax and to build the wings, etc. And depending on the organism, some of the genes are the same. In fact, some are very closely allied with other species. But they're turned on in different patterns. So this is a Drosophila fruit fly here and a wasp, closely allied to one another, both flying insects. And so this shows the pattern in the maternally expressed genes. For Drosophila in blue, the wasp in red, and both of them together are in black. And then gap genes down here. But you can see there's already a difference in pattern here. Hunchback, CAD, and NOS are about the same, but then this GT gene is different in that it only appears in the wasp, and the OTD is only in the wasp. But the OTD looks somewhat like the BCD in the Drosophila, where it turns on and its actions. That is, which genes it turns on and which ones it inhibits. By the time you get to the gap gene expression, there are further changes. So a lot of the genes, again, in black, are the same, and they're turned on in the same pattern. But then the red ones and the blue ones, some of them are the same gene, but they're turned on in different patterns. So the KNI is turned on in slightly different places in the wasp and the Drosophila, and then there's further changes here in the wasp where KNI is turned on anteriorly as opposed to Drosophila. So as these are affecting other genes and turning on and affecting the cells that are in those areas where those proteins are turned on, you get a slightly different body shape for each one of those. And remember, each one of these are turning on at a time when these are larvae and not adult organisms. And it's only later that the Hox genes are turned on to form the final body form for each one of those, where to put the legs, where to put the antenna, how long to make the antenna, if there's a stinger or not, etc. The Hox genes and proteins are members of a larger group called homeodomain or homeobox genes. Hox genes are only in animals, but this shows the fruit fly up here, nematodes all the way down into elephant sharks, zebrafish, and mice. And so these are the vertebrates down here, and up here are the invertebrates. But you can see that along the chromosomes, these groups of genes, Hox genes, are arranged in the same order. So from 1 to 15, some of them have up to 15. And they're always expressed in order as well, so that it's a very organized group of genes that are coordinately expressed in the animal, so that you get a correct pattern. You know, you get the head end, the tail end, you get the head in the right place, you get the legs in the right place, you get the tail in the right place, or the legs and the arms in the right place, etc. And not only are the arms in the right place, but the muscles are there, and the nerves are there in exactly the same place. So only by this precise order do you get an organized multicellular animal in this case. There are a few differences in that in nematode here, there's an inversion of 2 of those genes on the chromosome. And here in sea urchin, there's also been an inversion. And that then changes the body plan with respect to some of the other organisms. So you get a nematode instead of a fruit fly, and you get a sea urchin instead of an acorn worm, etc. Also note that by the time you get to vertebrates down here, there are multiple versions. And so this has happened because there's been whole genome duplication in the vertebrates. So originally there was only one here, like in the lancelet, only one copy. Then there was a duplication of the genome, so then you had 2 copies of everything in the genome, including Hox genes. And then there was a further duplication, creating 4 copies in sharks and reptiles, mice, mammals, and birds. But in bony fish, there has been another duplication. So originally there were 8 copies. Now you can read down here, and there's only 7 because one of those versions has disappeared, has gone extinct essentially, one of those gene loci. And working a little further back, as I said, the Hox genes are a subset of the homeodomain or homeobox genes. So that's this HD here. Those also appear in Archaeplastida, that is plants, fungi, and probably amoebae. Probably the slime molds have these too. And you can see that as the organisms get more complex, as they evolve, they had more and more versions of the Hox genes, and so their body plans become more and more complex. So for sponges, porifera, they're very simple. They essentially have an inside and an outside, and that's it. And this has gene loci. It only has one. It only has one gene, so it's very simple. And then going into sea anemone, they have a mouth, a gut, and a holdfast, something to hold them to the rock surface. And so they have more, but essentially these ones up here control the mouth and the gut, and then this green one down here, controls where the foot or the holdfast is. And so they still have only one or two, they have only one genetic locus, but they have between 2 and 5 genes depending on how complex they are. So as you work your way up in this direction, then here's nematode with more copies, more genetic loci, and then into echinoderms like starfish, more genes, just one locus still. But then you get into these vertebrates up here, first the tunicates and the vertebrates. And now there are more and more genes and more genetic loci, and especially in things like zebrafish, Danio. There are 7 genetic loci, originally 8, but it lost one. So now you have even more complex body parts, more complex body plans. So you can see that here. These are colored in the same colors as the Hox genes in the previous diagram. So here's a sea anemone, with a mouth, and this is would have the gut inside and then the holdfast. So you have just a few different versions forming this body plan. A little more complex in nematodes, as far as this is the embryo and then this is the adult worm, where those genes turn on and how they turn on the different body parts. And then into Drosophila here, which is more complex, and then into the chordates, where now there's a more complex body plan. These are fairly simple ones, but then you get to the more complex chordates, and in this one you can see not only one layer of the Hox genes turning on, but several layers turning on, which will form things like the epidermis and the neural tube, including the vertebrae, the spinal column, the muscles, and all of the other tissues to make a complete organism. And as I said, these are these homeobox genes or homeotic genes and homeotic proteins. They are transcription factors to turn on other genes. And so here's a homeotic gene. There's a variable region here, a homeobox here, and then another variable region at the end. And if you look at the protein, just at the homeobox itself, then these letters up here stand for specific amino acids that are well conserved in those homeobox or homeodomains of the proteins. Now, those are fairly well conserved not only in animals, but they're somewhat conserved all the way to plants. So they've been around for a long time, and especially this helix, which has a lot of these Ks. K stands for lysine. Lysine is positively charged. DNA is negatively charged. So you can see it fitting into the DNA. So it's attaching to the DNA because of that positive and negative charges, one on the protein and the negative charge on the DNA, on the phosphate groups. Two of the other domains also interact somewhat with the DNA, but they're pretty much there to orient that domain 3 or helix 3 that's attached to the DNA. And then the other parts up here are transcription activation domains. So those attract other transcription factors so that they can turn on or begin expression, transcription specifically, of those genes. And they will usually turn on an entire set of genes in a particular order in order to form an arm, a leg, a finger, a fin, things like that. So this is one example, and this was some data from a mouse forelimb and hind limb, but it could be a human arm and leg as well. So in the limb bud, HOX9D or D9 is turned on first, and then 9, 10, 11 a little later, and 9, 10, 11, 12, and 13 later. And then in the mature organ, then the forelimb or hind limb, you can see how these Hox genes are turned on and how they are forming those different limbs by turning on various sets of genes. So a shoulder will be formed and a hip will be formed when only HOX9D turns on a certain set, and then HOX9D and 10D will then turn on here to form the upper arm or thigh, etc. So that all the way down here where you get fingers and toes, it's 9, 10, 11, 12, and 13 that are activating other genes to turn on those fingers and toes. And it turns out that those genes, the Hox genes, are controlled by other elements. One is an early enhancer, and another one is a global control region. So those will exhibit transcriptional control in a coordinated manner, so that the early enhancer has its most effects on genes, Hox genes 1 through 4, and the global control region has most of its effects on genes 9 through 13, so that then you can get a blending of these two and you can actually change them from one organism to another to create slightly different limbs in this case. If you look through the different organisms, going all the way back to sponges and forward to mammals and birds, you find that in the beginning there were very simple patterns of these Hox genes. So they are transcription factors. The proteins are transcription factors. So they're turned on and they turn on another set of genes somewhere else in the genome. And then there were duplications of those, so you had more, and they started evolving into different functions. So now you get a head end versus a tail end. And there were more duplications and more diversification, so that now you had more distinction between the head and tail and the middle. Eventually there were some local control elements in some of the organisms, and so those would control also when those were turned on and in what order they were turned on. And then finally there were these enhancers, upstream and downstream enhancers eventually, that would start to take over control from these local control elements. So that by this time here, then you have both enhancers and global control elements that are controlling, are coordinating the transcription and expression of the Hox genes, which again are turning on other sets of genes to form different parts of the body. And eventually all of the local control elements lose their effects, and that function is superseded by the effects of the enhancers and global control regions. The story doesn't really end there. So the Hox genes are one part of the developmental plan, but there are other genes, in this case GLY3, so that the Hox genes here, if you have the wild type here and wild type GLY3, you get a fully formed foot, and this is for a mouse. And then you start losing, you get a heterozygote with one mutant copy of Hox, and you start getting less distinction in the fingers or toes. And then if you have a double mutant of Hox13, you get more of a club rather than a foot with toes. On the other hand, if you have totally normal Hox-D13, but then have either a heterozygote or homozygous for the mutant GLY3 protein, gene and protein, then you get more of those toes and they're ill-defined. By the time you have the double mutant, you get a club-looking structure, appendage, that has many, many bones in there that would eventually be fingers or toes. This can also be seen in evolution, so that this is an evolutionary tree on the bottom with the Hox genes on the bottom also. Here's copies gene 1 through 14 in the ancestral chordate. So that's a chordate leading eventually to mammals and birds. Here there's a duplication, so now there are 4 versions. This one is a jawless fish, and then up here the ancestral version with 4 copies is the jawed fish, and the first group off of that is the sharks, and you can see the pattern here. So you have many, many bones in the fin, and then a fin after that. And the colors of these bones also correspond all the way through to birds. So next you have another duplication in the Hox genes, and that's in fish, bony fish. And so you have even more bones, and still a fin, but you have more bones. And Hox-D13 is turned on only to small levels. So now you can see that you have a large number of bones and still a flat-looking fin. Along this line, there hasn't been that second duplication, so there's still just 4 copies. But you get into the lobe-finned fish, and there the level of Hox13 is increasing, so you get fewer bones. And so now it looks more like sort of a club with a fin extending off of that. And then into reptiles here, where you have a number of toes, but they're well defined as toes and not as a fin. And finally, further increases in Hox D13 to form fewer toes, or in the case of birds, the fingers and the wings, and longer bones as well. So then you get a well-defined hand, a well-defined wing, well-defined toes on the feet. And in fact, if you look at birds, they only have 3 toes. Most dinosaurs also had 3 or 4 toes. And so they had slightly higher levels of D13 so that they had fewer fingers or toes on the end of their appendages. So as I indicated in one of the slides before, there are several layers of these tissue layers with different Hox genes turning on and off. So if you look at sharks, chondrichthyes, and then ray-fin fish, lobe-fin fish, and eventually in tetrapods like us, these genes in different tissues are turned on in different ways. So these are neuronal tissues up here, and these are mesodermal cells, which will form the muscles. And so if you look in fishes, those HOX4, 6, and 9 are turned on higher in the spinal column and also in the mesodermal tissue to form the fin, the forefin. But to form the forelimb in tetrapods here, they're turned on lower in the spinal column so that the arm is formed further down on the body than the fin is in the fish. So if you had this pattern like fish, then your arms would be coming out just below your ears, right? They're coming higher in the spinal column. On the other hand, if fish had the human pattern, then their fins, their forefins, would be further back on their bodies. You note also that you have to have coordinated expression of these so that the muscles form in the same places where the nerves form, so that you get innervation of those muscles properly and the arms and fins work. So the next few slides are showing where these different things are turned on, these different Hox genes are turned on to form those different organs, different parts of the body. So here's neuronal tissue. This is in tetrapods where the neurons for innervating the forelimbs are here, hind limbs there, and in fish, the front fins and back fins here. In a tunicate, this is the head essentially and the tail, and then hemichordates, a different pattern there, and hexapods or insects there. So that's for the neurons. So then going to somatic tissue, this would be muscles and things like that in the arms. They're further back on the body for the arms and legs for a tetrapod, for the front fins and rear fins on a fish. A tunicate, and mainly these are showing where the tail of that tunicate is. There's some genes turned on further toward the head also. Hemichordates, a different pattern, and then in annelid worms, you have several different worms, and you note that some of these are sort of fading in and fading out because those genes and proteins have a greater effect in some places and a lesser effect in other places. So there's a more gradual pattern in some of these body plans than in tetrapods, well, vertebrates in general. And then going back a little further into arthropods and sea anemones, here are the arthropods, and you can see they have, in general, a similar pattern, but then there are differences in exactly where some of those genes are turned on and turned off in the body plan. So up here you have spiders and scorpions and centipedes, crustaceans, and insects. And then here's the simple pattern of a sea anemone. This is essentially the head and mouth. And this would be the body and gut in here, and then the holdfast down at the bottom. Here's a very simple chordate. That is, it has a notochord, and we have a notochord too, so we're chordates. But this is a very simple one, a very early tunicate called Oikopleura. And there's how the how the Hox genes are turned on in the epidermis, in the muscle, in the nerve cord, and the notochord. So you can see each of the cell type layers, each of the cell layers has a slightly different pattern for turning on and off those Hox genes. So you can get even more complex body plans. This animal's quite simple. This is pretty much the animal here, and then with a long tail with the notochord. So you can see that's exhibited here in that the epidermis has very few Hox genes turned on in the tail, also the ones in the muscle, etc. in that tail region. There's a bit more complexity up here in the main body, and this is the body. So there's the siphon, the pharynx, the esophagus, stomach, intestines, ovary, testicle, and then a small central brain, central ganglion. And so most of the body is here, and of course in us, our notochord forms early in development in us, and then kind of fades away as it's replaced by the spinal column, which of course is a characteristic of vertebrates. Okay, now I want to talk a bit about the human genome, that is of Homo sapiens. Well, Homo sapiens is quite variable, right? Genetically, you can have all sorts of different looks of people, but they're all able to reproduce, that is, form viable offspring. So you can go to any part of the world and form viable offspring with virtually anyone else in the world. But there's quite a bit of genetic variation. So when we talk about the genome of a population of organisms, or even a whole species of organisms, it's really it's no one's genome, really. It's the average genome. So you're looking at different versions of certain genes, so that when they do the human genome, it's really a mixture of many different humans to get that genome. And also humans have hybridized with other species. So it's true that Homo sapiens originated in Africa, but other Homo species were present in Africa, Asia, and other places. And so as humans started to spread, Homo sapiens started to spread right away in Western Africa. They hybridized with a different species of Homo, and no one knows exactly what it is. Maybe Homo erectus, maybe Homo neanderthalensis, but there's a hybridization event there, and you can tell by the different genes that group has. As they moved out of Africa, probably over 150,000 years ago, they then hybridized with Neanderthals, and that's been fairly well established. And then that group moved out through Asia first, and then moved into Australia, so those became the Aboriginals. However, there was another hybridization event here, maybe with the Denisovians, which were an offshoot of the Neanderthals, or probably with Homo erectus also. So now there's what's called an introgression, other genes coming from a different species. As they moved up into Europe also, there was another hybridization event, probably with Neanderthals again, to form a European population. And then finally there was this group, probably 14 to 15,000 years ago, that moved into North America, became Native Americans, and then continued down into South America, probably just one migration. And then a subsequent migration just a few thousand years ago of a group up here in Siberia that were already accustomed to living in cold temperatures, moved into northern North America, and those are the Inuits. So again, when you're talking about the human genome, then it's really a mixture of several different species, some of the genes coming from different species of Homo, and also they were moving into different regions at different times, so they've spread out and become very diverse. And so there's quite a bit of not only phenotypic variation, but there's some genetic variation in the human population as well. So here's the human karyotype and genome. About 3.7 billion base pairs of DNA, 23 or 22 somatic chromosomes, and then an X and a Y chromosome. And here's the number of genes on each one of those and the number of proteins that can be formed by each one of them. And so you can see we have approximately, and it depends on the study that you're looking at and how they define a gene, anywhere between 23,000 and nearly 30,000 genes. But we can form between 90 and 100,000 different proteins. So there's a lot of alternative splicing going on. And as I've mentioned in other videos, one of the largest genes, it's over 2.3 million base pairs in length, and includes, I think, 77 or 79 exons and introns. That's the dystrophin gene that forms a protein responsible for muscle movement. And when it's mutated, it causes the disease muscular dystrophy. And that's caused just by a single mutation in one of the coding sequences that creates a stop codon. And so the protein, when it's being translated, is short and does not function. So the muscles don't work. So within the genome, there are between 150 and 2,300 copies of ribosomal RNA genes. Again, variable on the chromosomes. 497 tRNA genes, not all expressed at the same time, of course. 97 SNO RNAs, which are responsible for processing ribosomal RNAs as well as some other RNAs, and more than 75 spliceosomal RNAs and additional small RNAs too. So a large number of small RNAs in this genome as well. In addition, the human genome has about 42% of the genome made up of transposons. And this includes the lines and signs, those highly repetitive regions as well. And about 1.5 to 2% consists of genes, about 10% non-coding RNAs, so more non-coding RNAs than genes actually. And then other parts of the genome, 48%, including quite a few introns and unknown sequences of unknown function. So remember when I was talking about plant genomes, especially the maize genome, over 80% of its genome was made of transposons, primarily retrotransposons. And fewer portions of the genome were made of introns. And so you had less alternative splicing in plants. Here now introns are a larger proportion, transposons are a smaller proportion, and there is more alternative splicing here. So plants have kind of expanded their transposons as a way to vary their genome. And in general, animals have increased the number of introns as a way to increase the coding variability, the number of different proteins that can be formed. And if you compare animals with a fungus here, then in general, about 25% of the genome is in common to all of those. And then if you look at worms, humans, and flies, essentially animals, then 68% of their genome is in common. And then you can work your way out to see some of the sequences that are unique, or the percentage of sequences that are unique in each one of those. So you see, they have quite a bit in common within this entire group, and especially among animals, with respect to the proportion of the genome that is similar. And another part of the story of genome evolution is looking at how chromosomes evolve. So this is a human chromosomes here, a human chromosome set here, and a mouse chromosome set here. And you can see from humans to mouse, both vertebrates, so they're evolutionarily related. You can see that there have been some rearrangements of the genes on the chromosome. The genes and gene types are pretty much the same, although the alleles are certainly different, so that then you get a mouse versus a human, and then the Hox genes are turned on differentially, so you get differences there. But you can see here on chromosome 1, part of that is on chromosome 1 here in a mouse, and part of it's on chromosome number 4 as well, and a little bit on chromosome number 8. And then you go to chromosome number 2, and there's part of that on the mouse chromosome 1 and 2, and some others in here on chromosome 6, and a little piece on chromosome 17, and chromosome 12. So that and 11 also. So pieces of that are all over the place, even though those genes are the same genes that you find in humans. So this is similar to what I showed you for the grass family, as far as some of the chromosome synteny, that is where you find analogous pieces of the chromosome. Those genes have moved around during evolution. A couple of things that haven't changed so much are the X and Y chromosomes. So those genes pretty much stay on the sex chromosomes, because those determine the sexes, and that's a pretty important distinction among those organisms. Okay, so this is the last video in the genome series of 4 videos, and just talked about eukaryotes, large genomes, and remember all of those things, that those control mechanisms and things that were happening in virus genomes all the way back there are also occurring here, but there are many, many more of them. So I couldn't cover the details of as far as the expression of those genes and control of expression of those genes, but those same things are happening here, but just to a much, much greater extent. Okay, so that's it for this video. Thanks very much.